and I see Chuck Behorn out at the Grassmere Gardens, and worked with a lot of great people out there, and a wonderful relationship was developed for a lifetime, I say, Andrea. Well, then I find out that time passed quickly for me, and I find out that she's now a doctor at Vanderbilt who is doing and has been doing significant research on the COVID pandemic. And so, Andrea, I would like to have you share with all of us your antiviral program. Is that what? Okay, and tell us about that and lead into your presentation for us. But thank you for taking your time and being with us tonight.
It's my pleasure. And I actually have everything incorporated into one presentation, including who I am and you know, what I do. So I'll just start sharing my screen.
Oh, Andrea, I'm going to interrupt you for a minute.
Yes.
I see Rich Shivey down there at the bottom of the screen, so I don't want to leave him out of being the team that you got to play and work with.
I'll also add, while we're pausing, I'm going to mute everyone except Andrea so that we don't have any background distractions, okay?
Okay,
so,
sounds good. And I'm not muted, right? No, everyone can hear me. Can everybody see my screen? Yeah. Actually, yes. I only need one yes.
Yes.
So the title of my talk is
You're muted.
Now I'm muted. So okay. I was automatically muted along with everybody else.
Yeah,
but now I'm
sorry
muted.
about that.
So, no, it's okay. It's okay. That's an effective way to mute everybody else. Okay. So my name is Andrea. Most people know me. You know, I normally look like this. Let's see if I can have a pointer that is a little bit more visible right here. Probably one of the last times most people saw me was at the Urban Garden Festival, and I met Buzz the bee over there. I spent most of my time looking like this over here in a laboratory working at Vanderbilt University Medical Center. So my life, along with everybody else's life, changed dramatically about a year ago when the pandemic started. And unlike most people in the world, we were kind of excited about that at first, because we've been studying these viruses, coronaviruses, for about, well, our lab has been studying it, not myself, but the lab has been studying it for about 20 years because they are a large family of viruses that circulate in animals and in humans. And so when we heard about this mysterious viral pneumonia in China, we were thinking, sounds kind of like SARS-CoV, there was an outbreak in 2002. Let's see how this develops. Well, this is our lab over here, and I have to credit everybody. We are about 10 people. There are about 10 people in our lab, mostly women, as you can see. And we are located at Vanderbilt University Medical Center in this building right here. And we all work for this guy, Mark Dennison, and he's the one who's been studying coronaviruses for more than 20 years. He's also a physician, he's a pediatrician, and so this is how he went on rounds not so long ago, wearing the clown nose. And I had to remind him that the mask goes over the mouth and the nose, but he took it off. So like I said, we've been studying these viruses for years, and until recently, whenever I would be in a conversation with anybody outside of my lab, basically, people would ask me, what do you do for research? And I would start talking about these coronaviruses, really interesting viruses, very abundant. People's eyes would just kind of glaze over and the subject would sort of be changed, you know, about a minute into me telling them about it. These days, everybody wants to hear about it. And that's one of the reasons why I'm here tonight. So over here, this is something that most of you are probably not used to looking at, but this is what we call a phylogenetic tree. And that is a way for geneticists to illustrate the relatedness of different species, or in this case, strains. So coronaviruses make up a large family. Every single name on here, even though it's kind of a cryptic name, it's a different coronavirus. They can be subdivided in different groups, and then by different subgroups, and then you have the species. Now, most of these you've never heard of before, and I don't blame you. There are 3 on here that you might have heard of before, and they are all in yellow boxes. So SARS-CoV, it was the first coronavirus. This has sort of hit the news. It caused an outbreak in China in 2002, 2003. And that was really the first time the world met a coronavirus that could really do some harm. It caused a lot of death, but fortunately it was contained within China. It didn't really spread outside the country. Then in 2012, we had an outbreak of Middle East respiratory syndrome coronavirus, or MERS-CoV, which is actually an ongoing outbreak. It's also coronavirus from a different subgroup. It actually causes a lot of mortality. About 35% of people that end up in the hospital, they die from it. Fortunately, MERS-CoV doesn't spread very well from human to human. It's mostly spread from camel to human, and that's, I think, why some of us have never heard of it before. But at that time, that was a reason for us and others to start thinking about, okay, we have nothing to treat these coronaviruses. We have no antivirals, we have no drugs, we have no vaccines. We need to do something with it, because at some point we're going to get an outbreak of a coronavirus that causes a lot of disease and mortality and spreads easy from people, from human to human, so between people. And that happened last year, sooner than we had hoped, but it happened. So SARS-CoV-2 is the causative agent of COVID-19. So COVID-19 is the disease, SARS-CoV-2 is the name of the virus. Other human viruses, human coronaviruses, are shown in these red boxes, and they cause common cold-like symptoms. And you've all been infected with one of or more of these multiple times in your life. You just didn't know it, because common cold, we don't really worry about it too much. Like I said, these viruses, they circulate in animals, and every once in a while spill over into humans. So a lot of these coronaviruses have been found in bats. That's why I have a bat over here. If you can see on this phylogenetic tree here, every name that starts with a BT, it's a bat coronavirus. And on my next slide, I show you the 7 strains of coronavirus that we know of that circulate in humans. Over here on the right are these low pathogenic CoVs. We call them CoVs, is short for coronavirus. And they cause common cold-like symptoms, but they originated either in bats or in mice, and they moved into humans probably via an intermediate host, a host that humans are more often in contact with, such as cows or maybe llamas. These highly pathogenic CoVs that emerged more recently, such as SARS-CoV, MERS-CoV, and the source of the current pandemic, SARS-CoV-2, also have an intermediate host. For SARS-CoV-1, it was the pangolin, or sorry, the raccoon dog or palm civet. These are animals that live in the east. Dromedary camels for MERS-CoV, and perhaps, we don't know for sure yet, perhaps the pangolin, which is a very unusual looking creature. Although there's a chance that maybe it was directly transmitted from bats to humans. So there's actually currently a group from the WHO who has been led into China to thoroughly investigate where did SARS-CoV-2 come from, because we know its most closest relative circulates in bats, but we don't know if there are other intermediate hosts for it that we need to watch for when we go and control this virus. So a question I get often when I show all of these coronaviruses is, why did SARS-CoV-2 cause a pandemic while other coronaviruses did not? And we don't know the exact answer to that just yet, but it probably has to do with the fact that it spreads easily from human to human, and that unlike SARS-CoV, the first one, SARS-CoV-2 infections are often, in about 80% of infected people, they're mildly symptomatic, and the symptoms are kind of, sore throat, maybe a little bit of loss of smell. It's not really very distinct, so people may not think much of it and go about their life rather than isolating. More recent numbers have shown that about 59% of all the virus is spread before people even become symptomatic, and some people don't even become symptomatic. So if you have a lot of people that are spreading the coronavirus that don't even know they're infected and don't take adequate precautions, you can see how this virus can spread very easily through a population. And it will be hard to control because it will be hard to identify the people that spread it and isolate them. So coronaviruses, this may not be new to you, but I'm just going to go over it for completeness sake. They spread through the air and via direct contact. So the way they spread through the air is by droplets, which are produced if you talk, breathe, cough, sneeze, sing. And they can travel a ways. They can travel for meters or yards. They don't really linger. They usually eventually drop down to the ground. But some of these portion of the coronaviruses are spread via aerosols, which are tiny, tiny droplets. And they can linger in the air. So if you go into a room where an infected person has been breathing without a mask freely, there's a chance that you'll breathe them in and you'll get infected. A third route is contact, so direct contact through what we call fomites. So it's basically, I say I'm infected, I touch my mouth, then I touch the door handle, the next person comes and opens the door and then touches their mouth. That's a way of transmission as well. Now that way of transmission was very much kind of a focus in the beginning. We were all spraying down our groceries. I don't know if we all were, but I was disinfecting everything. But it turns out that that actually only accounts for about 10% of all infections. Now you should still wash your hands often. Be very aware of where you put your hands, but bleaching of the apples is probably no longer necessary to contain the spread. Also, don't go around the grocery store, lick the door handles or anything. You know, be careful. Keep doing what you're doing. But it's pretty. It seems like right now we're mostly focused on the droplets and the aerosols for routes of transmission. So David asked in an email, you know, a lot of new and you know, current master gardeners would like to start volunteering again and working in the garden. And I don't blame you. I think we all want to get out and go on with our lives. Maybe some tips for gardening safely in groups. I say that it's possible. You just have to take your precautions and really understand how to protect yourself. Some tips that I have, of course, you know, if you want to be 100% safe, get vaccinated and never leave your house. But I wouldn't recommend that for mental health's sake. So try to find a nice balance where you're aware of the risks you take. But yeah, do stuff. So stay home if you notice symptoms. I would say avoid carpooling because if you're carpooling with somebody else, they may not realize they're infected. They may breathe out virus particles and you're in a trapped space. These aerosols, they can linger. If you must carpool, not everybody has a car, not everybody has a way to get places. I would suggest you wear a mask and roll down the window to improve the ventilation in the car. For the same reason, you might want to avoid entering enclosed spaces. I'm thinking garden shed, greenhouses, especially when they're poorly ventilated. And then in terms of gardening, you know, outside, just be aware of how far droplets can travel typically, and you know, how they're produced. So if you cough, you can produce droplets that probably come down, you know, somewhere between 2 and 6 yards. You know, think of yourself as a circle. If you sneeze, it can go up to 6 yards. That's or more, which is a long way. If you're just breathing, talking, exhaling, droplets probably, you know, drop somewhere between 1.5 meters or yards and 2 yards. So I would say if you garden outside and you keep your distance, preferably more than 2 meters, don't spend, you know, a lot of time talking to each other. Talk to the plants instead. You should be relatively safe. You're outside. It's a big space. Aerosols can diffuse and go away. The best thing you can do though is wear a mask. I will always recommend wearing a mask. as if you can handle it. I know some people, they get claustrophobic and it's really uncomfortable. Try different masks, see what works best with you, because you not only protect other people, you also protect yourself if you do so. So gardening, keep your distance, wear your masks. Oh, and I would try to avoid sharing garden tools, because we just talked about contact transmission through fomites. You know, one person uses the shovel, next person uses the shovel, touches their face. You just want to be aware of where you put your hands. So now I'm going to go into a little bit more science. I'm going to tell you about coronaviruses, what they look like at a molecular level, and how they infect the cell, and how they basically change the cells to make factories to make more of themselves. So a coronavirus, just like any virus, it's an inanimate object. It's not alive. It can't do anything as long as it's not within a host cell. It can just sit there. It will be stable for a little bit in the environment, depending on what surface it's on, at most a couple days. It needs a cell in order to do what it does. Coronaviruses like lung cells or cells of the upper respiratory system. And this is a schematic representation of a coronavirus particle. It's made out of RNA. Its genetic material is RNA. We have DNA. This virus has RNA. And it is protected by a protein shell. And around that is a lipid envelope, but you can't see that one in this picture. What's interesting, what I'm going to point out about this protein shell, it's made up of multiple types of proteins, but the green one over here is called spike. And you may have heard of spike, because spike is actually really important for the virus, because it mediates the attachment of the viral particle to the host cell. So as I said, viruses can't do anything until they infect the host. They need a host. So what they do is they bind to a receptor, which is basically a protein that your host cell in your lungs or your upper respiratory system expresses. It's called ACE2. And it binds with this spike protein, it binds to that receptor, triggers fusion, and via that mechanism is basically taken up by the cell. Once it's in the cell, it releases its viral genome, so the RNA gets released from this protein shell, and it gets picked up by the host replication, host translation machinery. So this is basically, your cells are always translating proteins to stay alive and to do their functions. The virus takes advantage of that. So it has the right signals so that the host thinks that it's its own RNA that it needs to go and translate. Makes proteins, and then it makes more pieces of itself. It moves through the cell and assembles into new viral particles, and that is one viral particle can turn into thousands in a matter of hours. So it's very efficient. Then once there are many viral particles, they'll start exiting the cells and looking for new uninfected cells, usually neighboring cells, but it can also end up in the bloodstream and go to different organs. For SARS-CoV-2, it has been found not only in the lungs and the upper airways, has been found in the kidneys, in the heart, other areas of the body it can infect. Now, we are in the business of stopping this. So how do you stop this process? Well, there are many ways you can stop this process, actually. You can stop pretty much every single step of this process that the virus needs to make more copies of itself. You can stop the binding, you can stop the uptake, you can stop the translation of viral proteins, replication, so multiplication of the genome, you can stop the exit of the virus. So ideally, we'd like to target all of that, but we don't have drugs that do all of that necessarily that will stop this virus. In fact, it's actually really hard to stop this virus because it's always kind of one step ahead of the research, as it seems. It's very good at evading these kinds of drugs that work for other viruses. They don't work for coronavirus. So until recently, we had no drugs that treat these infections. And that's why 7 years ago, our lab said, okay, if we ever have a pandemic that's caused by a coronavirus, we are in trouble because we have nothing to treat sick people with. And that's kind of what happens. Because antiviral drug development, it works, it takes a lot of time. So our drug, or our lab about 7 years ago, started working on trying to find antiviral drugs that work against these viruses. And we decided to target this part of the viral replication cycle that is to make, where the virus tries to make more copies of its own genome. And we did that because it's a very conserved process. The virus cannot really mutate around that. So if you put drug pressure on a virus, often it will undergo mutations to find a way out of that drug pressure. Now, it can't really do that for these types of antiviral drugs that target this area of the replication cycle. And so we had about 4 years ago, we had developed 2 drugs that worked really well for other coronaviruses. And so when this viral outbreak happened, we said, okay, we're ready. We got 2 drugs ready to test. You know, if they work against this new coronavirus, we can move them into clinical trials and test them on humans, which that hadn't been done yet. But first, we needed the virus to test it on, which is, I think, you know, kind of duh, but we were watching this happen in China and then in Europe, but nobody in the US had come down with the infection yet. So we had nobody that we could culture the virus from. But then on January 21st, 2020, we had our first case of COVID-19 in Washington State. Fortunately, this person wasn't very sick, but they had recently traveled to China and they were very aware of this new disease, and they presented to their health care provider, and they were sampled and it was tested, and indeed it was this virus. So samples were sent to the CDC, and we happen to have friends at the CDC. One of my friends works there in the lab that first cultured this virus, and they figured out a way to culture this virus, make more of it, which, you know, that sounds counterintuitive, but we need more of it in order to study it. So they cultured it in a very safe way and then sent it to us on February 6th of 2020. This box arrived, which contained the virus. So we were very excited and we were ready to go. We had our drugs ready to go. We had our lab set up. You know, we'd been working on these viruses for many years, so we knew how to work with them safely and what precautions to take at Vanderbilt. So we tested our antivirals, and this is probably one of the most complicated slides of the entire presentation. If you zone out, just come back to me after this slide is done. But I just want to show you about the 2 drugs that I just talked about, how they work, because it's pretty nifty, I think. So the viral genome, so the genetic component of the virus, is made up of RNA. And RNA is made up of 4 different building blocks that are arranged in different orders: a U, a C, an A, and a G. The drugs that we have tested and that we have developed are iterations on these building blocks. So basically what they are, they look like these building blocks, but they're slightly different. And what they do is basically, the virus doesn't know this. They, you know, using the host cell machinery, incorporates these drugs into their genome. And then all of a sudden, the virus can't replicate anymore. It basically it stops replicating. And you can see that in this figure where the SARS-CoV infectious titer goes down when the drug concentration increases. And this one works by actually incorporating and causing errors. So subsequent rounds of replication, the genome gets all messed up and it's not a correct copy anymore, and it cannot make any more proteins, and the virus cannot infect any more cells. So that's one drug. The other drug is also a modified nucleoside. And this one is called Bremdesphere, and you may have heard of that one because that one is actually the first and the only antiviral that's been approved by the FDA for use in COVID-19. This one was previously tested against Ebola, and so we already had a lot of human safety data. That's already been tested on humans. We knew what doses were tolerable, and so this could be moved into clinical trials quickly. We just had to show that it worked against the virus, so we did. Over here, you can see, again, viral titer drops when the concentration of the drug increases. And unlike mutagenesis, this one causes chain termination. So as soon as it gets incorporated by the polymerase falls off, all replication stops. So in the lab, it is very effective against SARS-CoV-2. So that was exciting that worked. Both of these worked. So Remdesivir in the summer was approved by the FDA. The other one is called Molnupiravir, and I apologize for these names. They're very complicated. It was picked up by Merck and is now entering phase 3 efficacy trials. And we have high hopes for this drug because it's an oral drug, unlike Remdesivir, which has to be administered by an IV. So only people in the hospital really have access to it. Whereas Molnupiravir is a drug that can be used as a pill. So you could, if you are in a high-risk group, a high risk for developing severe disease, you can take it as a pill while you're still in a mild phase of the disease, while you're still at home, and it could knock it out just like Tamiflu could. That's our hope. Now, this has to still be shown in the large-scale efficacy trials that are coming. But if this works, then it's really going to be a game changer for treating patients. Another method of inhibiting virus would be monoclonal antibodies, which you may have heard of before too. And these are basically components of the immune system that are produced in the laboratory. So this is a natural way for the body to defend against a virus. So you can actually generate those in the lab too by injecting a mouse or generate them from the blood of people that have been infected with the virus and have produced a neutralizing immune response. You can then produce these monoclonal antibodies, test them to make sure they neutralize, and make many of them and purify them, and then use that as a drug. So if somebody is infected, they can actually be infused with this drug. And President Trump received this treatment when he came down with COVID-19. So what it does, basically, these antibodies are, they bind to the outside of the virus, and keep the virus from being able to attach to this attachment receptor on the human cell. So it's basically kind of like they run pass interference. I don't know if that's the correct football term, but basically they just block the virus from being able to enter the cell in the first place. So you really nip it in the bud that way. We have 2 approved monoclonal antibody therapies right now. One is made by Regeneron and one is made by Eli Lilly. They also have impossible names that I'm not going to try to pronounce, but they really seem to make a difference in terms of efficacy. A single IV infusion can reduce hospitalization rate of high-risk individuals by 3-fold. So it's not a silver bullet, it's not a magic treatment, but it's something, and it's better than nothing. And it seems to be one of the most effective treatments we have so far. And I just want to show this graph over here because it's really important when we think about antivirals and other treatments to think about the timing of these treatments. And I think one of the reasons Remdesivir is approved and is somewhat effective, but it's not a great drug. And the reason probably is because people have to wait until they get into the hospital to receive it. And it's usually for any antiviral. By that point, it's probably too late to treat it effectively with an antiviral. And that is because COVID-19, there are 3 stages roughly of infection. There's early infection where the virus enters, replicates to high titers. And then some people, never get sick and are fine and recover by just staying home. But then some people get shortness of breath, low oxygen. This is the place where maybe there's not, the virus starts sort of disappearing, but the host immune response starts really, is triggered and starts acting up. So this is massive inflammation where the lungs, basically, there's a cytokine storm and the lungs start filling up with fluids. This is where people get shortness of breath, low oxygen, and they might present in the hospital. Severity of illness is shown here with this black line, so you can see it go up. Stage 3 is, and I have some pictures to illustrate kind of what these stages would look like. Stage 3 is hyperinflammation stage, where people have to be intubated, and many people don't survive this stage when that happens. So for an antiviral, antiviral drugs, you really want to use them in this early stage of infection, especially in high-risk individuals who have the potential to really get really sick, because by the time this happens, it's too late to really suppress the virus. It's more a job for immune modulators to suppress the inflammatory response so that the body has a chance to heal itself. Now that is kind of an overview of where you would want to treat with what. The best thing to do is really to prevent infection in the first place. So this is our big boss again over here, showing 2 thumbs up. Before infection, behavior and vaccines are really the ways to prevent infection. So I want to go into that now. I'm not going to go through this viral replication cycle again. I just want to show you different ways to prevent infection is by social and physical distancing. So meet as few people as you can while staying sane. If you meet people, try to keep your distance, wear your mask, wash your hands. And as soon as you have access to a vaccine, consider getting it, because I think this is the way that we're going to get out of this pandemic. So I want to talk to you now about our role in vaccine development, because you know, since we were one of really 5 labs in the country that worked on these viruses before this pandemic hit, we've been in the spotlight. A lot of people have come to us for expertise and the ability to actually do experiments that needed to be done to show that drugs are effective and that vaccines work. So we've been involved in the United States of America government. They launched a big program called Operation Warp Speed. And this was an initiative that the government launched to accelerate the development of vaccines and therapeutics. But they've been mostly in the news because of their success in vaccine development. A lot of money has been poured into it. Some of it went to the National Institutes of Allergy and Infectious Diseases. There's a vaccine research center who had been studying coronavirus vaccines for years. So they had a lot of expertise. A small company that's really up and coming called Mrna and us at Vanderbilt University Medical Center, we've been working with the VRC, the Vaccine Research Center, for years. So we had existing relationships. So when this vaccine needed to be tested, they came to us, which was really, really, for me, exciting to be able to be involved in that. I want to talk to you a little bit about what we did in terms of vaccine development and testing. But before I tell you about what we did, I have to explain to you how vaccines work. Otherwise, it's not really, you know, it's hard to understand exactly what we did and why we did it. So really the goal of vaccines is to prepare the immune system and to bolster it for the event that it sees an infectious agent, such as a virus, such as SARS-CoV-2. There are different effector mechanisms. So the immune system will build an immune response against things that it sees that are foreign. And these things could be pollen, you know, hay fever. These things could be infectious agents, anything it sees, and sometimes even things that, you know, are part of your own body in case of autoimmune diseases. But let's talk about it from the perspective of a virus. So normally when you're infected and you've not been vaccinated, it takes your immune system a little while to get activated, often about a week to really get going, which for some viruses, that's just too late. You're dead by then. Ebola, you're dead. You don't have time to wait around for your immune system to start. It will actually react by making an immune response that is both cellular, so T cells, and humoral, which is antibodies. And this initial immune response, it's not very robust. It's, you know, it happens, but it leaves to be desired. It's not super protective. It will come up, and then over time, it will just sort of taper off. And then immune memory is formed. And that's really where the good stuff comes in. Immune memory, these are cells that remember seeing this first infection. They have been optimized to target whatever causes first infection, so that when the second infection comes around, it's ready to go. It doesn't need much time to come up and to start attacking this virus and neutralizing. So it's both a faster response and a more robust response. Now, fortunately, with vaccines, we don't have to wait for this first infection. We can just vaccinate, and tell the immune system what to look for and what to remember. And then later, when the virus comes around, it's ready to go and it can respond quickly without, any risk of disease. Most vaccines you've had for childhood infections or even just the flu vaccines, they're often not what we call sterilizing. They will protect against infection. They will reduce disease severity, but they're not 100%. So a lot of vaccines, when they are designed and when they're made, they're supposed to, the goal is to keep you alive, to make you survive the infection, not to completely make you not notice the infection. So when the COVID vaccines were developed, this was sort of a goal, and that's important to keep in mind. Of course, a secondary goal of a vaccine is to reduce viral spread in the community overall and to build long-term immunity. So that if you, ideally, some vaccines will give you immunity for 10 years or more. That would be nice. Not all vaccines can do that, though. So vaccine testing, let me go over this too, because a lot of people don't realize what goes into vaccine testing. There are different phases of testing. It actually starts with preclinical work, which is the laboratory work that is often not recognized, but can take many years to develop the idea of what could be a good vaccine, to do animal experiments, to test, is it safe for animals? Does it actually induce a robust immune response? Could this be something that could work in humans? When all that looks good, that's when phase 1 trials happen. Phase 1 trials for the Mrna vaccine started in March of last year. A small group of healthy volunteers, so 45 people, young, healthy, had no other problems, signed up. I'm just in awe to receive this vaccine that nobody had before, for which we didn't know it was safe. So if you talk about heroes, these people are the real heroes here. So these people signed up to get the vaccine. They tested different doses to see what would be a tolerable dose. One dose was not very tolerable. These people didn't feel so good, but the other 2 doses were fine. People didn't notice anything really. So initial safety of this vaccine looked really good at these lower doses. These people were still actually tracking them for long-term side effects if any of those come up, also for long-term immune response. But by the time a few months later, the FDA had looked at it to see, is this really, does this look good in terms of safety, in terms of dosing? A phase 2 trial was started, and shortly after, a phase 3 trial was started. And as you can see by these orange people, every phase enrolls more people. People become more diverse in terms of age, demographic, and then the phase 3 trial is really when you start testing, not just for safety, but also for efficacy. So in the phase 3 trial, that was one that started in August, and I was actually a part of that here at Vanderbilt. Vanderbilt was a site. There were different sites all over the country where people got enrolled that wanted to be a part of this study, from all different demographics, all different ages. Some people had other comorbidities that were, like other diseases that, we wanted to know if the vaccine would be effective. Half of these people received a vaccine, and half of these people received also an injection. It looked just like vaccine, but that was a placebo, what we call placebo. It was a salt solution, physiological salt solution. So nobody knew what they got. I didn't know what I got. The people that injected me didn't know what I got. It was blinded. And that's really important, because you don't want to, have the people that think they got the vaccine to change their behavior to all of a sudden, take more risks or anything. You want to know, does this vaccine work? This phase 3 trial, it's now turned into a phase 4 trial. That was not shown on this original graph. I added it myself. All of these people now are actually still followed to make sure that it's safe in the long run, and that and to determine how effective these vaccines are. So this is kind of what it looks like to be in a clinical study. And I know some of you might have wanted to sign up for this if they had a chance. I was lucky that I knew the people who were running this trial, and I was able to get in. On study day 1, this is Mrna vaccine. You get a vaccine, they draw your blood, and this is kind of your pre-immune blood. You have not seen the vaccine, you have not seen the virus before. 4 weeks later, you get your booster, which is again a dose of the vaccine. They again take your blood, they take nasal swabs. You know, I don't know if any of you has gotten tested. It's not very comfortable, that they need to know, are you positive or not? Which, you know, that's important to know. And then every so often, you get your blood drawn again to see how does the immune response develop and how does it last. So in our lab, we received these blood samples from the first 45 people that signed up for this phase 1 trial. And our question that we wanted to answer is, do the antibodies, so these immune components that neutralize the virus, how well do they neutralize the virus and for how long? So what we did is we took a portion of this blood, it's called serum, that these immune, these antibodies are a component of, and we diluted it and we mixed it with a set amount of virus. This is live virus. This is in the lab. You know, I have my hood on. I have my own air purifier. This is highly contained. We mixed it and then we put it on kind of a lawn of cells in a petri dish. And what the virus does is, if it infects a cell, it will spread to the next cell and to the next cell and to the next cell. And after 3 days, you can actually see clearings in this layer of cells that we call plaques. And we can count those. We actually physically count those with our hands, our eyes, and a Sharpie. And then we score and we say, okay, so, a dilution of 1 to 20 resulted in 8 plaques. If we dilute it a little bit further, there are more plaques. So this is kind of a dose-dependent increase in how well the virus is able to infect. This was really exciting for us because this was done in May of 2020. And I still remember seeing this first assay where we saw that the serum of these people that are signed up for this study was very, very highly neutralizing of the virus. So these people that received the vaccine were able to neutralize the virus really well. We had never seen such a thing before. So we were ecstatic and we couldn't tell anybody because this was highly confidential. If we were to tell the press, then everyone would go and buy stocks from Mrna and everything would be a mess and we could end up in jail. So we kept our mouth shut until the press release came. The stock shot up and exactly what they said was going to happen was going to happen. There was a lot of excitement for our data that we generated in our lab, and we're still tracking those people. So when they get another vial of blood drawn, we see, okay, how well does it still neutralize after so many months? Because this neutralization is an indication for how effective the vaccine is. I get a question a lot, how long does immunity last? And we don't know yet. Because, March of 2020, that's only 9 months ago. We're not really that far into these analyses yet. So far, from what we see, there's still a lot of neutralizing antibodies. Naturally, these neutralizing antibodies, they will wane, and it needs to be replaced with this long-term immunity that you saw in my diagram before. And we don't really know when that's going to happen yet and how well that will happen. Now I want to tell you a little bit about these mRNA vaccines because they're new. It's a new platform of vaccine development, and it makes a lot of people nervous because, it's something they've never heard of before. So let me walk you through this. So this is the coronavirus again. It has these spike proteins that it needs to attach to the cell. So if you block these spike proteins, virus can no longer attach to a cell, can no longer infect. So researchers, whenever the genetic code became available, was made public by researchers in China in January, they designed what this vaccine should encode in about 4 days because a lot of money had already been poured into such an approach for MERS-CoV, so that other virus that's circulating in the Middle East. The Vaccine Research Center had been working on that for years. And it had already shown that those vaccines were effective against MERS in animal models in primates. So it knew, okay, if we offer this piece of genetic code and we encase it in a lipid coating and inject that into people, then these lipids nanoparticles will mediate the delivery of this tiny piece of genetic code from the virus that encodes for the spike into the cell. The cell will make proteins that look just like the proteins that a virus would produce when it were to infect the cell, except there's no virus here, it's safe. Then the immune system will recognize this as foreign and produce antibodies against it. and will produce antibodies against a form of the spike that's highly, that's highly what we call immunogenic. So it's actually recognized by the immune system. And these antibodies can then neutralize the virus. So that's kind of how that works. Often this is done, other vaccines, the way they work is they will inject a sick version of the virus that doesn't replicate well, or a different virus that expresses spike protein on another virus that's harmless and replication incompetent. Sometimes people will just inject proteins and have the immune system respond to that. In this case, it's a little bit more fine-tuned. It's this genetic code that that the cell can use to make the proteins as if the virus was infecting, and it's a very kind of precise way to do it. mRNA is safe because it's non-infectious, so there's no risk of, oh, did the virus get inactivated correctly? Did the agent that was used to inactivate the virus, is that harmful in some way? None of that. It has no preservatives. It has no adjuvants. Often when vaccines trigger side effects, it's often because there are adjuvants or components in the vaccine, like aluminum, that actually trigger the immune response. mRNA vaccines don't have that because they don't need it. mRNA does not integrate into the host DNA. It just gets broken down by the cell. After it's made these proteins, the cell will degrade it just like it would a natural mRNA by normal cellular processes. It's a minimal vector, as they call it. So the immune response is focused only to this protein that the immune response actually needs to see in order to neutralize the virus. It's not misdirected to all kinds of other proteins that, the antibodies won't, even if they bind to them, they won't be able to neutralize the virus. And as I said, the technology has been in development for more than a decade. So yes, it's new in humans, and it's a rapid way to make a vaccine, but it's not that new, really. It's been studied extensively. Now we have two of these vaccines are now approved, or they have an emergency use authorization received by the FDA. One is made by Pfizer, and one is made by Moderna. Moderna obviously is the one that we worked on, but it's very similar in terms of how it's made, what it's made up of, as the Pfizer vaccines. And they have very similar efficacy. So they're both about 95% effective in preventing disease. It's really important that people are open to receiving these vaccines because the way vaccination works and the way it can protect the whole population is by what we call herd immunity. So if everybody is immune to this virus, either by vaccination or natural immunity, the virus has no place to go. It can't find new hosts, and so it can't make more copies of itself, and it will disappear from the population, or at least exist in very low levels. Right now, it's in the population at very high levels. I have this is an animation that shows you the differences between percent vaccination. This is 0%, no one has had a vaccine, versus 95% of people have had a vaccine. So if only 25% of the people, get vaccinated, it really doesn't have much of an effect on overall virus spread throughout a population. Eventually, most everybody will get infected. If half of the people get vaccinated, you drop it down a little bit, but you can see there's a lot of red still. 75% is pretty good. 90% is really kind of what we're going for. So, I know there are more vaccines that are coming on the market that are still in trials that have promise, like the Johnson & Johnson vaccine. We'll have data of that vaccine soon. That one was made by a more traditional platform, so it's not an mRNA vaccine, it's an inactivated virus, or a replication deficient virus, which is what people are more used to. So even if you don't get this vaccine, get a vaccine, I would say that. So takeaway points from my presentation, and I'll go into some frequently asked questions. It's already almost 7:30, sorry, I'm talking a long time. Coronaviruses are a large family of viruses of animals and humans. They spread via the air and via surfaces and infect the airways. Vaccines against coronaviruses will prevent infection Antivirals drugs will stop the infection, and immunomodulators will halt disease progression. Different types of drugs will target different aspects of the disease, and the timing of administration is important. Vaccine development and testing was accelerated thanks to Operation Warp Speed. A lot of logistical, because so much money was poured into it, the operation, or everybody was allowed to work together to make this process go as fast as it can without cutting corners. And in July was when we didn't even know if these vaccines were effective yet. Companies were already given money to start producing these vaccines so that if they turned out to be effective, they would be ready to go by the millions, as opposed to waiting. Normally companies are very risk averse. They will wait to see all the data and all the efficacy data until they even start pouring money and producing the vaccines. If that had happened, we would have had to wait another year before we had a vaccine ready to go. So that was one of the reasons why it went so fast. mRNA vaccines also, because we already knew so much about how to make an effective mRNA vaccine, is why these two mRNA vaccines are the first ones to be available now. It's important that they estimate that we need about 80 to 90% to be vaccinated to generate that herd immunity that will keep everybody else safe, even people that have not been immunized for some reason. And the main goal of vaccination is really to protect against infection and to prevent the severe disease. So maybe, do you want me to just keep going, or shall I take questions now? I'll keep going. So I'm just going to go into some quick vaccine FAQs, and I have included some pictures of my garden because obviously I've been kind of busy this past year, so I've not really had much time for volunteering and all that jazz, but I was able to get out in my own yard, especially in the early morning hours. I would sometimes just take half an hour to myself, just look at my flowers, take pictures, enjoy it, because it was important for me too to stay healthy mentally as well as physically through this entire process. Well, a question I get a lot is, should I get vaccinated? Well, I think from my presentation now it's clear that yes, you should get vaccinated as soon as it's your turn. The temporary discomfort of the vaccination itself is nothing compared to the acute and long-term side effects of contracting the disease. So a wholehearted yes. Well, what about me? I'm young, healthy. I'm not really at high risk for severe disease. Should I still get vaccinated? Yes. Yes, you should still get vaccinated because low risk is not no risk. Plenty of young and older, otherwise healthy people get really sick and die of COVID-19. Many people with mild symptoms, even if you're young and healthy and you didn't even have to go to the hospital, you caught the disease, you tested positive, you've had it, they will still feel very tired and have maybe shortness of breath months after they've recovered or they're no longer positive. So this can be a disease that kind of lingers. So really, it's going to make you feel crappy for a while. Long-term symptoms include shortness of breath. Some people have heart rhythm problems, brain fog, they can't concentrate, and other neurological symptoms that we're still learning about. There's no reason not to get the vaccine. The vaccines are effective and safe. I have received my own, dose in August and September, and I have no long-term side effects. Another common question I get is, should I still get the vaccine if I already had COVID-19? And that's really a good question because presumably you might have some immunity against it already. What we've learned so far is that natural immunity to the virus is often not very robust or long-lasting enough to prevent reinfection and disease in the long term. So it's very hit or miss. Some people who have maybe had a more severe form of the disease might have formed a good, robust immune response that's long term. But other people that may have had a mild form may not have the necessary immunity to actually prevent reinfection. And that's why we see reinfections now. People that have had it in the summer got it again in December and think, what the heck? I've already had this. I thought it was done with this. That's one reason why it's important for everybody to get the vaccine, and you're being advised to do so. Pretty picture from my yard. This is a monarch butterfly on my milkweed. Can I get COVID from the vaccine? No, you cannot. The vaccines are mRNA vaccines. It's just a small portion of the virus. There's no virus. It was not made in the presence of virus. There's no accidental virus that got into it. It cannot give you COVID. Now, it will generate an immune response, and your immune response might make you feel like you have something, but it's very temporary and it will go away, and then you'll be immune. This is me over here. How can I be sure the vaccine is safe? This is after I had my first shot. They took my blood, they gave me my shot in my right arm because I'm a lefty. This was also on World Left-Handed Day. How can I be sure it's safe? That's a really valid question. The vaccines, both Pfizer and Moderna, were tested on more than 77,000 trial participants of different ages, race, and health status. These people were closely monitored. I can tell you, I get phone calls, apps, text messages all the time checking in with me. You know, do you have any change in your health? Do you feel anything weird? Like tell us everything. No safety issues were observed. Also, a lot of people say, okay, that's all nice, but how about long-term side effects? Because what if 5 years down the road I turn purple? Nobody will know because these vaccines have only been tested for 9 months. We don't know about the long-term side effects yet, and that's true. We don't know. We can't say 100%, you know, there are no long-term side effects. But what we do know from the history, and we've been vaccinating people for many, many decades, is that side effects of vaccines are very, long-term side effects are very rare. So there are very few vaccines that we know, if you can actually count them on one hand, that seemed to be safe initially, but down the road turned out to not be so safe. And that has been studied extensively. Why is that, and how can we avoid that in the future? And that has to do with the type of immune response that's mounted in response to these vaccinations. It's basically a suboptimal immune response that causes issues in the long term. We know from studying these immune responses from this vaccine that is not the case for this vaccine. This vaccine mounts the right kind of immune response, the one that is protective and not harmful. So that is what we know. But what if I have condition X or Y? Should I still get the vaccine? Get that question a lot too. One thing that I think is for the mRNA vaccines, you should ask yourself, do you have a history of anaphylaxis due to any cause? If so, make sure after you receive the vaccine, you stay for observation for 15, but ideally 20 minutes after receiving your shots. All the people or the places that you will be able to get your vaccinations, they've all been instructed to have EpiPens on hand, because in a very small subset of people, they will actually have an anaphylactic reaction to a vaccine. For the COVID-19 vaccine, right now the data show that it's 11 people out of 1 million have showed some kind of anaphylactic shock, which can be treated with an EpiPen. So that's why these EpiPens are available. So if you have a history of this, talk to your doctor first. I'm a PhD, I'm not an MD, I'm not allowed to give you medical advice. Talk to your doctor about your specific health condition and whether it's safe for you to get the vaccine. This anaphylactic shock in these 11 people, you know, out of the 1 million people that have been vaccinated, or at least that proportion of people, it's probably due to a component of the vaccine that is used to protect the mRNA. It's called PEG. It's a difficult name, polyethylene glycol, and that is under investigation why that causes these reactions in that very small percentage of people. So tell your doctor if you have allergies, if you have a fever. All of these reasons are maybe reasons to either delay or not get the vaccine. So please, you know, make this an open dialogue. I expect in most cases your doctor will say, don't worry, you're fine. Your risk of getting COVID is much greater than for this vaccine to have a negative effect. Question. Do I still need to wear a mask after I get vaccinated? And that's a very, you know, a very abundant question. I get that a lot because now people get vaccinated and they're all, excited and they can go and do their things again and they can hang out with their friends that are also vaccinated. Do they still wear a mask? I would say for now, keep wearing your mask because you might be protected, but the person you're interacting with may not have received a vaccine yet. And right now we know from the data that the vaccines are very effective at preventing symptomatic disease, but we don't know for sure how well they prevent transmission of the virus. So maybe people that are vaccinated could still be infected and produce some virus. And that is all stuff that still needs to be measured. That wasn't measured in the first phase 3 trial that we have the data from. So likely it will prevent transmission as well, but we cannot say that for sure yet. We also don't know how long the protection will last. So maybe this is a vaccine that we have to get another shot, and like a flu vaccine, we have to get a yearly shot. Maybe it's, it's more like one of our childhood vaccines that will last for 10 years. I mean, that would be great, but we just don't know until we hit those years after we've been studying this. So protect yourself, protect others. Asymptomatic infections, you can still spread the virus. What I just said, efficacy was 95%, not 100%. So bottom line, as long as the virus is abundant in the community, it's important to just keep protecting yourself and others and be aware of that. And hopefully come this summer, maybe end of the year, we can all be together again. We're all vaccinated and we can all just, have our dinners together, go to our concerts, do the things that we like to do. Recently, you may have noticed in the news, there's a lot of panic about new viral variants. So variants that have arisen in the UK that seem to be more contagious. There's a South African variant that seems to be, maybe more resistant against the immune response that's built against the original virus. Will the vaccines protect against these new virus variants? So far, some of these studies have been done and they show that the vaccines protect against these new variants too, because immune response is very diverse. It's not, if a small piece of the virus spike changes, there will still be antibodies that target different parts of the spike that are the same and will still neutralize the virus. So far, it looks like we expect that the vaccines will still be effective against these new variants too. But it's possible that just like the flu vaccine, we have to update the vaccine every year, make it slightly different so that we can adequately protect against the current variant that's circulating. Hopefully not. Prairie coneflowers in my front yard. These are my favorite new addition to my yard. Actually, I pocketed some seeds from Madison, Wisconsin, from our family visit there, and put them in my yard, and they do really well in Tennessee too. They bloom for a long time and then they rebloom again, and they seem to stay alive above the ground for throughout the winter. So I'm expecting some nice new flowers again in the spring. So let's see if we had them all. Okay, just briefly, who's eligible to get the vaccine? We're currently vaccinating based on age, so 75 and older, and based on risk, healthcare workers. So we're in phase 1A, 1A2. Some counties, surrounding counties to Davidson County, have moved into phase 1B, which includes school and childcare workers and 70-plussers. If you want to find out what phase are you in your county, either contact your healthcare provider or go online. There are two good websites that I found where you can find this information. For people that live in Metro Nashville, you can go to the Metro Nashville Health Department website and click here or call to make an appointment, get on the list so that they contact you when it's your turn and vaccine is available. Or you can go to the Tennessee State Health Department website and click on your county. So I've clicked in this picture, I've clicked on Davidson County, and it will give you information about what phase we're in, if vaccine is available, what phone number to call to get on the list, or you can go online and request an appointment by filling out your information online. So those are two places to go to. Now I want to just finish before I take questions with some lessons I learned during the pandemic. I don't know if Diane is on, but she gave me some seeds of these beautiful Venus poppies that grow at the demonstration garden. They grow in my yard now too. They're just gorgeous. Number one is, since I didn't have much time to garden this past summer, I just decided to sprinkle a bunch of zinnia and cleome seeds. And really that did the trick. It kept me, supplied with color for many, many months, and drew in some pretty butterflies too. Volunteer gourds can be a benefit of not weeding. So I stopped weeding and I found this gourd in my garden. So that was quite exciting. And the garden is really a magical place if you take the time to truly experience it. And this was a moment that I had in the morning. It was foggy outside and I thought, oh, it's kind of cool light. I'm going to take some pictures of my zinnia. So I crouched down to take a picture of these zinnia. And then I saw all these deer in my backyard. And it was just, I just snapped this picture by chance, really. It was beautiful. So I welcome questions now. If you are bold enough to ask a question, right here, you can do it. If you'd rather talk to me in private, this is my cell phone number. I'm happy to take your questions if you want to call me, or if you're more of an email person, you can always email me as well. I'm happy to answer any questions because I think that's important. Thank you for listening.
We've got a bunch. Thank you. This is
Okay.
awesome. We've got a bunch of questions. I'm going to just start at the top. So Tom Coulter wants to know, do you have recommendations on effective masks? Germany recently required particular masks, the FFP and the FFP2 for their citizens. Do you have any recommendations?
I don't, to be honest. Okay. Multiple layers is always a good thing. Surgical mask, they have some kind of a charge that will actually repel virus from even landing on your mask. So they're just the disposable blue ones that you see around. Those seem to be fairly effective. I would just make sure, that they fit well. They don't have, they don't have too many gaps. And they're comfortable enough that you don't have to keep touching them, because I think when people touch their face a lot to adjust their mask, they inadvertently can actually transfer the virus onto their face, and then it might get into their eyes or their nose or their mouth. So those will be my my general questions, but I don't have any specific brands or anything that I can recommend.
I think this was Connie Vargas, because it says John. She said, excellent presentation and thank you. And you talked a little bit about this, that about the new strains coming from South Africa. Are people working on new boosters or anything, and is this and then you also talked about, will this be a yearly vaccine like the flu vaccine, which nobody's sure yet, but is anybody looking at boosters?
Yeah. I think actually I might be, since I'm still in the trial, they might actually do a booster on all of us if it turns out that the immunity wanes. My blood gets analyzed every so often, and the neutralizing antibodies and the long-term immune response. I think these trials will just keep going for a while. These people that have been enrolled will stay in the trial for a while to see how long protection lasts, and based on those data, I think the researchers that make these decisions will decide on whether or not to give a booster. In terms of these variants, they get tested in the lab. So basically, what people do is, and we haven't, unfortunately, not been involved in this part because it requires more people than we have. Really, we only have a few people in our lab that can do this work. But what they do is they test the immune serum from people that have received the vaccine to see if it neutralizes these new variants. So it gets done that way to see, okay, compared to the original virus, how well does the immune serum neutralize these new variants? And that's being done on an ongoing basis. And the South African variant is the only one that has been shown to be neutralized a little bit less well by the immune serum from people that have received the vaccine, but a little bit less well. There's still a lot of neutralization. So it may not even, make a difference at all in effectiveness of the vaccine. It's just on a laboratory scale. It seems to be a little bit of a drop in neutralization.
Okay. Byers is asking, what's the deal with people who have allergies? And I'm guessing that's, are allergies a problem with getting vaccines, and does it make it better or worse? And I'm sure the sneezing doesn't help.
Most allergies shouldn't affect it. If you're unsure, talk to your doctor.
Do you know of any positive effects of vitamins, herbs, anything on suppressing COVID? Have you heard
No.
anything? No?
Nothing has been tested in a trial yet, but it looks like levels of vitamin D actually correlate with disease severity, but it's not sure that actually treating with vitamin D is going to do anything. I think keep your vitamin D levels at a healthy level. It might help you stay healthy. Yeah.
Okay, good to know. Deb Wilcox was asking, how are you notified or kept up to date on new information, especially on the variants?
Oh, Twitter mostly.
Hilarious.
No, we talk to our colleagues regularly, and whenever something new breaks, there's quite a buzz about everything. So I read the literature. A lot of stuff will end up in a newspaper before it's even officially in the scientific literature. So I'm only half joking about the Twitter thing. There's a large community of virologists that shares information on Twitter, and I pick up a lot that way too.
Okay. Then there's appreciation for your native wildflowers supporting pollinators in your gardens.
Thank you.
And many thanks for your work and for the presentation. Carolyn Turner said that there's 2 people in her building that had COVID and received an infusion. What would that have been?
It's either Bremdesphere, probably, or these monoclonal antibody therapy.
Okay, yeah, you talked to us about that. Vaccines for children, are we going to, how will we reach herd immunity if children vaccine is down the road?
Yeah, so children were not included in the original trials, mostly because of consent issues. So children often need their parents to consent for them, and it's complicated. Right now, Pfizer is doing large-scale trials on children to look at efficacy in children, and Mrna, I think, has started them too. So hopefully, in the next few months, we'll know more about that.
Okay. Do you have to get your vaccination in your own county? Can
That's
you go outside?
Good question.
Okay.
I don't know, but it's worth a try. Yeah, I mean,
Okay.
whatever you can. Cheat a little.
Can I hop in and say, this is
Go.
Jennifer. I'm going to Macon County tomorrow. I'm a 1B teacher and I live in Davidson but work in Robertson, but I put my name on the waiting list in a county that had a 1B vaccine and they called me today. So, and they specifically asked me where I lived several times and they had no problem with that.
Wow, that's great.
Good to know. Yep. Lots of thank yous. Terrific presentation. Thank you for the information. Susan's asking about sharing the presentation with friends. Do you have a problem with that? We've made a recording and we're going to put it on our website. And Susan, you should be able to get it from that. If not, reach out to me. This is Karen Brown and I can get you the link.
I can also I actually made a PDF of this and I can put it in the chat if you want. Oh no, I can't. Well, I can send a PDF of it too if you want.
Okay, would you send that to info@mgofdc.org?
Sure. Yeah.
Okay. My husband is getting his, lots of people talking about they don't have to be in your home county. Joan Clayton Davis was asking, does the lab do presentations to minority groups to help address problems of trust with vaccines? And if so, can groups contact the lab for a presentation? For example,
I am trying to reach out to
health ministries or associations.
I would love to give presentations to those groups. Please contact me if you have anyone wants a presentation. It's one of my focuses now, and this is why I'm here tonight. I wanted to do this
awesome.
to reach people. So yeah.
Joan, call her,
Yeah.
Joan. Somebody else is telling us you keep bees and quilts and you make gorgeous quilts.
Thank you.
So,
yeah, I have many hobbies. My bees swarmed this year. I couldn't take care of them adequately.
Oh.
But I still have one hive left. So maybe next year when I have more time, I can pick that back up.
Well, thank you. Carol or David, you want to wrap up? We've gone through all the questions.
Well, I'll just mention a couple of things. This is so informative. This is a pandemic no one's experienced this in our lifetime, and everybody on this planet is affected by it. And I heard on the news that if every country is not vaccinated, almost up to 9% plus, these viruses linger and linger, and they just don't go away. So it's I'm honored that I know Andrea, and I know the rest of our association is honored that you do such important work. And what year did you go through the class?
I'm 2013 certification.
Gosh,
Yeah.
you're an old timer then.
I know, it's been a while. Yeah.
I'm glad you had time to work. Everybody has struggled with their gardens here, I believe, too. But I appreciate more than ever being a master gardener, but just the good work you do as a human being for all human beings, worldwide.
Well, I've just felt really blessed to be able to, be there right in the midst of it and make a difference, because I feel like, while other people were sort of, bored in their houses, I had something, I had work to go to every day, and I knew why I was doing it for the first time kind of in my researcher's life. I've been a researcher for 20 years, and sometimes I was kind of wondering, why am I doing this? I've not had that thought at all this year. It's been very rewarding.
Very good. I'll turn it over to Carol.
Oh, I just have to echo everything everybody said about thanking you. This is one of those unselfish acts. And we know that you've spent many hours, many days, many months in everyone's behalf. So what an unselfish act to come here and then to tell the rest of us that you're looking for opportunities to spread the news. It's emotional. And just thank you. I think this was a premier presentation. And what I mean by premier, it was so darn informative. So thank you from the bottom. I'm going to say again, from all of our hearts. And
you're very welcome. And like I said, please email, call me. Also, if you have some suggestions on how I can make this better, just let me know.
Good.
We'd like to, let's see, I at one time saw that we had somewhere around 40, between 40 and 50.
Great.
And we need to do our part now and see what we can do to get this presentation of yours out to others. There are roughly, they're close to 300 of us. And so we've got a little bit of our work to do to see what we can do to spread your message, which is get vaccinated and wear our masks.
Yeah, that's right. If you learn nothing else from this presentation, that's my message.
Great.
Well,
thank you so much.
Have a good evening, everybody. Thanks so much for
Thank
staying
you
so
for
much.
so long.
It was wonderful.
Good night.
Bye-bye.
Bye.